Hi everyone, welcome to today's event. I'm Shifa. I'm a second year studying infection and immunity at UCL, and this session today is hopefully going to shed some light on the programme and answer any questions that you may have. Our speaker for today will be Professor Richard Milne, who is the Programme Director of this undergraduate programme and a Professor of Virology in the Division of Infection Immunity. Thank you for coming. Hope you enjoy. And Richard, over to you now.
Okay, thanks very much, Shifa. Let me just get this going. Share. Right. Good afternoon, everyone. A very warm welcome to this talk. I'm really pleased to see you here. As Shifa just said, I'm Richard Milne. I'm a professor of virology here in the Division of Infection and Immunity at UCL, and I'm also the program director for the BSc Infection and Immunity that we run and that you are interested in. What I want to do today is give you a sort of brief intro to the Division of Infection and Immunity at the University Department and the discipline, what infection and immunity is all about. And I want to do that by giving you a sort of little intro into immunology, and then talk about some recent scientific discoveries that have been made in this department around COVID, actually. And the reason I want to talk about science is because in the end, that's what this program is about. It's about doing science and becoming familiar with research and the current thinking in this amazing field. And then I'll change tack and talk quite briefly, actually, about the BSc program, because everything you need to know is on the prospectus. And then we'll have a chance for some questions and answers. And I would encourage you to ask questions about the science as well as the program, because in the end, as I said, this is about the science. I'm guessing that you're here because you think you might be interested in doing this degree. And I think that's great. And right at the end, I'll give you some advice on quite generally about how to choose a degree and what you should be doing. But I think this is quite a specialist program, and I'll come back to that. But just keep in mind, if you're trying to decide whether this is the thing you want to do, as opposed to a much more general biosciences program, if you enjoy and are interested in and excited by what I say over the next 15 minutes or so, then probably this is quite a good choice for you. If you're not that interested, if you think what I'm talking about is old hat or a bit boring, then this definitely is not the right degree program for you. So hopefully one way or another, this can help you come to a decision. So we're a department within the medical school. I'm actually sitting at the moment in the Cruciform building, which is pictured on the right side there, which is on the main UCL campus in central London. And in this department, we have about 40 senior staff members we call investigators. And each of those investigators runs a research group. And some of those research groups are quite big, 10 or 12 people. Some of them are much smaller, 2 or 3. About a third of those investigators are clinicians, so medical doctors. And one of the, I think, quite cool aspects of what we do in this division is that the research that we carry out really does go from the laboratory bench, the very fundamental basic questions about biology of infectious agents and the immune system, right across the the sort of span to very clinical bedside intervention trials, treatment trials, and so on. And that's a great environment. If you want to study infectious diseases and the immune system, this is a good place to do it for that reason. So most of these investigators are working in either fundamental immune cell biology, how does the immune system actually work, or what happens when it goes wrong, so autoimmunity, when the immune system actually inappropriately targets you as opposed to some infection, or immunotherapy, which is a very exciting recent development where you harness the immune system and redirect it so that it becomes a therapy. And this is transforming cancer treatment, for example. Then we have people doing work on virology, viruses, I'm a virologist, and microbiology, so the study of bacteria. And we're really based over two sites, the Cruciform building I've already mentioned, which is here at the main campus, and the Pears building, which is this very swish brand new building opened a couple of years ago, which is at the Royal Free campus, which is one of the medical teaching hospital centres that's associated with UCL. That's about 15 minutes up the Northern Line tube from here. And this building, I mentioned the bench to bedside concept. This building is particularly interesting because you'll see it has two sort of layers to it. This part, I hope you can see my pointer, is the lab and the research staff office. But this bit with the big windows is actually a hotel for patients who are taking part in trials of innovative new immunotherapies. And they stay in that hotel and they are kind of a captive population. And I think that's a unique facility in Europe. The building is next door to the hospital. There's a great big hospital next door to here, and I'll show you a photo of that in a little while. So that's the division. I want to talk about our discipline and why it's important. And to get you thinking about this, I've got these pictures. So obviously this is the planet Earth. And if you consider all life on Earth, right? So everything that you've encountered in your biology lessons, the whole breadth and complexity of life, from the very basic single cell organisms right through to humans and whales and giraffes, whatever, and plants and everything. Essentially everything that's captured on the tree of life, which is what I'm showing over here. Think of all that and now reflect on this. Every single living thing on this planet is infected, in most cases with multiple agents. So that's a remarkable situation, because what it means is that infection is a universal challenge to life. And I think it probably has been that way since the origin of life. I suspect that viruses and other infectious agents are a kind of inevitable consequence of the existence of cellular life. And if you accept, I'm sure you agree with this, that in the end, it's not good to be infected, then you can see that this universal challenge is going to be a really profound pressure for adaptation and evolutionary change in all of these life forms. And what we see is that every living thing has some kind of system to protect itself from infection. And we call that an immune system. And so the immune system in a very real way is an inevitable consequence of the universality, the ubiquitous nature of infection. So although the degree sounds a bit niche, actually I would argue that it is dealing with really fundamental, universal biological characteristics. And I think that's important. So we are a division of infection and immunity, and they often go together. And I just want to make a point about why that is, and make that by showing you some clinical pictures. So I'm sure you would all recognize this. This is chickenpox. This is quite a cranky baby, and he's cranky because they're really itchy and scratchy, these vesicle rashes. But he's going to be fine. He'll be okay in a few days. This is also a case of chickenpox, but this is very attenuated. You can see there's hardly any spots here. It's just one little area of swelling here. This is also chickenpox, and this is a gentleman who has had a kidney transplant. And of course, if you have an organ transplant, you have to take drugs to suppress your immune system to stop it rejecting the transplanted organ. And that's amazing that we can do that, but there is a price to pay, and that is that you have a slightly enhanced risk from infectious diseases. And that's what's happened to this gentleman. And this is a life-threatening situation, although it is treatable. So the point here is interesting. What's the difference between these three? Because I've told you they've all got chickenpox. It's the same virus in all three cases. And the difference is in the infected person, and in particular, it's in their immune status. They're each in a very different immune status as far as this virus goes. So this person was vaccinated, and so when they encountered the virus, they already had an immune response, and it controlled the virus very nicely. So you have this attenuated infection. This little baby is healthy, but had never seen the virus before, so gets classic chickenpox. This gentleman, as I've said, has been therapeutically immune suppressed, and so he cannot handle the virus, and it replicates and spreads in an uncontrolled manner. So what we're seeing here, these different presentations of the same virus, illustrates that the way the disease presents, and we call that pathogenesis, isn't just a feature of a virus or an infection or a host. It's the consequence of the interaction between those two. And where that interaction occurs, things get very interesting, because that's where the pressure to evolve an immune system happens. That's where the sort of biological pivot around whether the patient survives or dies occurs. And that's really why this is such an amazing and rewarding subject to be studying. So I thought I would give you now a very short sort of intro to the whole concept of the immune system. And these are slides from a lecture that I give actually to some first year students. So this gives you a sense of what you would get if you came to UCL. And I'm not an immunologist, and I have quite a simplistic perspective of immunity. I think of the 3 R's, and I find this very helpful. So the first thing the immune system has to do is recognition. It's got to recognize that something has gone wrong. And the sort of classic historical description of this that you may have heard of in your biology studies is distinguishing between self and non-self. Actually, it's much more complicated than that. But in this case, here's some cells, and this pink one might be infected by a virus, or maybe it's a tumor cell. And the immune system has to be able to tell the difference between this one and these ones. And if you think about it, that's amazing, because the immune system has an almost infinite capacity to recognize stuff. Anything that you encounter, it can recognize, and that's the fundamental basis of the whole system. So then it has to respond. And in this case, I've made it so that this might be a T cell, and it's killing this virus infected cell. And that's good, right? Because that will get rid of the virus. What you don't want to do, however, is have the immune system running out of control. And so it has to be regulated. And regulation really is at the crux of the whole thing. It's the regulation of the immune system that really controls what goes on and what gets killed and what doesn't. And in fact, quite a lot of the immunotherapies that I talked about at the beginning are really about playing with regulation, making the immune system recognize something that it wouldn't otherwise recognize, or stopping it from recognizing something that it shouldn't recognize. So if you were an infectious disease, or maybe even a cancer, you would want to avoid this. And the way you would do it is what I've written here. You could avoid being recognized, so you could change regularly. You could actively evade the response, so come up with some way of skewering a T cell, or you could disrupt and derange the regulatory processes. And there are lots of examples of all of those scenarios throughout infectious diseases. And some of them are really important, and some of them are ongoing in a way that really impacts on us as patients. But in terms of how this plays out in the sort of disease, inflammation is a word you might have heard of. When you get sick, you have swollen glands. For example, your neck might swell up, or under your armpits. That's your lymph nodes and making loads and loads of T cells to counter an infection. That's like a classic immune response. And then there are scenarios when things go wrong: immunodeficiency, immune system stops working; autoimmunity, as I've mentioned, when it attacks you; or cancer, when some of your cells are allowed to grow out in a deleterious way because they're not controlled by the immune system. And when we repurpose the immune system, we can address that. We can give you vaccines to prevent infection or maybe cancer, and we can use immunotherapy to redirect this aberrant immune response to solve the problem. So that's the kind of broad framework of what immunity is all about, and people in our division are doing all of this stuff. What I want to do next is give you a much more detailed story, one single story of some work that's been done and is ongoing, actually in this building upstairs. And this is about COV2. So obviously, as virologists, we've had an interesting few years. And back in January 2020, virologists asked themselves, what do we do? You know, how can we contribute to this situation? And my colleagues upstairs in this building decided to essentially carry on doing what they'd been doing with other viruses and apply it to COV2. And this is Lucy and Lorena working upstairs in one of our high containment labs early on in the COV2 experience. And this picture on the right is actually the very first set of pictures we got of this virus when it arrived. And these are cells. The blue sort of circles are the nuclei, and these are actually airway epithelial cells, and they've been infected with the virus, and we can detect the virus with this green stain, and you can see quite quickly it spreads through these airway epithelial cells until almost everything is infected. This was pretty exciting, actually, you know, to see a new virus in the lab that is causing this pandemic. So I want to tell you a particular story that kicks off, and you may remember this, or you may have blotted it all out, or you may be sort of suffering from COVID fatigue, but I hope that you'll find this interesting. You might remember around sort of Christmas time of 2020, a new variety of COV2 appeared, and it was originally called the Kent variant because it was first detected in Kent, but it was renamed the Alpha variant, and it seemed to be better at transmitting from person to person, and it seemed to cause more severe disease. And so what the guys in the lab did, first of all, you get a new variety of the virus. First thing you do is grow it in the lab. And here's some data. This is a figure that they produced. And this is what we call a growth curve. So you infect cells, as I've just shown you, and you wait for 24, 48 hours, and you measure the amount of virus that is produced. Here we're measuring actually viral nucleic acid, viral genomes, and it increases exponentially over a pretty short period of time, actually. But the thing to note on here is that the alpha virus, the new one that transmitted better and seemed to cause more severe disease, is no better seemingly at replicating than the original COV2. So how do we explain that? That's an interesting observation. You'd think that it would replicate better, wouldn't you, if it's transmitting in the wild more effectively? So I'm going to explain to you, I'm going to give you an explanation for this, but I just want to give you some background first. So I'm assuming you know a little bit about viruses, and viruses are these crazy things that aren't actually cells. They're this whole separate biological class of replicative but non-living entities, but they need cells. They use cells and they turn them into factories to make more viruses. And so just as I was talking about earlier when I showed you the picture of our planet, cells have evolved antiviral defenses. So most cells actually are quite resistant to viruses. And one of the pivotal defenses is a protein called interferon. You might have heard of interferon. And it's what we call a cytokine, which is a protein that's released from cells and then triggers other immune responses. And this protein, interferon, is produced when a cell senses that it has been infected with a virus. And it's released and it triggers all the other cells in that area to express a whole repertoire of antiviral defenses. And so it arrests the virus infection. And viruses, you will not be surprised to hear, evolve to evade these defenses. And so you have this amazing kind of churning cyclic relationship that occurs throughout all virus infections throughout evolutionary time, where cells respond, viruses evade, cells respond, viruses evade, and that goes on infinitely. So one of the first things that the guys upstairs did, having got this result, was ask what's actually going on with this interferon response? Is that different? And they did a cool experiment. So they actually measured how much interferon was produced when each of these different viruses was introduced into some cells. And what I'm showing you here is actually a measurement. They're not measuring interferon itself, they're measuring the messenger RNA that encodes interferon. And this is two separate experiments with the original virus, the Wuhan strain, and you can see this is actually how much induction there is, and it's quite substantial. But a really remarkable observation is this one. When they then looked at the new variant, the alpha, look at that, it was an order of magnitude less good at inducing interferon. So what this means is that this new variant has been selected to be able to evade the cellular responses. So it's not inducing anywhere near as much interferon as the original virus. And so it has an advantage. It can replicate more effectively, it's not suppressed. And we think that that is a big part of why it was able to spread more effectively and transmit and emerge and really take over from the original virus. It's all down to the very, very specific capacity of the new variant virus better to evade the cellular responses. So when I said, and I showed you that picture of the planet and said this stuff was going on all the time, this is an example of that. And this is really important because it's this sort of change that has governed how this COV2 pandemic has progressed. And I'll show you that in a moment, but I want to make the point that what I've just shown you, those graphs, that is real scientific data. This is not some made up stuff to entertain students. This is a paper that the guys published, and this is in Nature. This is the top science journal, and I've actually highlighted. There's the graph that I just talked about. And in fact, there's one as well. So what I talked about is like a tiny part of the main message, but you can see the title here is exactly what I've just said: Evolution of Enhanced Innate Immune Evasion by COV2. So this was pretty exciting, and it got a lot of interest and publicity. But as you all know, since then, there have been other variants. Alpha, the Kent variant's long gone, it's ancient history. And we've had, you might have heard of Delta, you definitely will have heard of Omicron. And so a good question would be, what do those variants do in terms of interferon? And I mean, I can't ask you, but if I'd asked you, what experiment would you do? The answer might be, well, just do the same experiment that I've just shown you. And here it is. So here are the different, this is just the different Omicron viruses and the Delta virus, and they're all growing quite nicely. What do you predict? Well, given what I've said and everything I've said about the evolutionary pressures here, what you'd predict is that all of these variants would have enhanced their ability to evade interferon. What do you get? You do that experiment again, and you see exactly that. The early Omicrons induced quite a lot of the antiviral response, but the later Omicrons, that is diminished, and they have adapted to be better able to evade this response. So this kind of research and getting involved in this is a big part of our approach to our BSc. And obviously everyone around here has been working on COV2, and this is just a small subset of the papers that have been published. But I'm showing this because I want to make one particular point, which is that these three actually have as co-authors students on our BSc program. So within a year or so of this pandemic kicking off, we had BSc students co-authoring COV2 publications. And that's where we want you to be. We want our students to be taking part in real science, really getting to the state of the art. And that's pretty exciting. So I've focused on COVID, but obviously it isn't just about COVID. You know, this business of infectious diseases is massively important. You might have heard about Mpox, it used to be called monkeypox. That just came out of nowhere. And I think we still don't fully understand why. There is an outbreak of Marburg virus in Africa at the minute. Marburg is closely related to Ebola, only I think it has, in this sense, a higher mortality. That is pretty terrifying. But there are experimental vaccines and treatments that are being evaluated. I picked this out of the other day, this urgent warning. This is about avian flu. Did you know there was a massive epidemic of avian influenza at the moment, worldwide? We've never seen anything like it. And of course, avian flu is the source of human flu pandemics. So we have to watch that very carefully. And you might have heard over the last year or so, late last year, there was an unexplained sort of outbreak of hepatitis in children. And that was worked out and explained and linked to a virus, an adeno-related virus, amongst others by colleagues in UCL. And these stories, I just picked these out kind of randomly. They're all over the last couple of weeks. So this stuff is really important and really relevant. Slightly tangentially, but since I assume you're all interested in this stuff, I want to refer you to this podcast. So Chris Van Tulleken, who is the presenter, actually did his PhD in this building in our lab, but he's a TV science presenter and also a doctor across the road. But he made this podcast about viruses that jump from an animal to a human, and they're fantastic. And I really, really encourage you to listen to these. And this is just quoting from him. He says, wherever you sit on climate change or welfare, conservation, inequality, these are engines of pandemic threat, and change is not happening fast enough. So climate change is driving pandemic emergence. And he says this, and I totally concur with this: either we dramatically reduce our impact on this planet, or a virus will jump and do it for us. And as we know from the pandemic that we've just experienced, that is not pretty. So I guess my point is that infectious diseases and immunity are pretty important and will continue to be pretty important for the foreseeable future. So that's our discipline and some of the science that we do, which you would better get your teeth into in a big way if you come here. I want to sort of finish by giving you a quick overview of the actual program structure. I mean, I guess you've seen this because if you're interested, you'll probably have looked at the prospectus. But my pitch is that we want to present our research and current research and use that as a tool to give you a broad insight into infection and immunity as a discipline. And you know, a big part of science is curiosity. You've got to want to find stuff out, because the business of scientists is finding stuff out. It's discovery. And that is a very creative process. So I really emphasize curiosity and creativity as sort of the pathway to discovery. And I want our students to sort of, it's a bit Orwellian, isn't it, to say that I'm going to change the way you think, but I want our students to start to think like a scientific investigator. So that means not just accepting what you're told, assuming that the textbook is true, but challenging, asking, why do you say this? What is the evidence for that? What are the data that support this? And we will definitely train students to do that. So it's a 3-year BSc program. The first year really is, it's about how the human body works. So, and that's at a cell level and at a systems level. And the different modules that you take are in this grey box here on the right. And they kind of mirror the medical school curriculum, which makes sense because we want to teach our medical students how the body works as well. The first year is a joint first year with a number of other programs in the medical sciences faculty, but they're all non-clinical programs. These are not programs that are going to train you to be a doctor. And the way these are delivered is called blended learning. And it's kind of old hat now because everyone's been through the pandemic, but rather than just having you roll up and sit in lectures the whole time, they get you to watch short video presentations or online activities, do that, and then attend the in-person aspect so as to get more out of it. And Shifa can tell you more about that if you're interested. So that's quite broad. The second year zooms in a bit. There's more of a focus on infection and immunity, but still really giving you a grounding in molecular medicine and basic cell biology. And then in year 3, really everything is on the table. And it's really about research, your research and our research. You do some core content on the left, and you choose 2 of these optional modules. And we really teach about what we do. We don't cover everything. There's no parasitology, for example, because we don't have any parasitologists. We're not in the business of reading a textbook and then regurgitating that to you. We're interested in research-led teaching. So you will hear about the stuff that we're doing. And I think that's the attraction of this. And that's where you want to be, right? If you're studying science. So what are the outcomes? What would you gain? Well, you definitely know about how infections and the immune response impact on human health and disease. The health aspect is really important. It's easy to think of the immune system just as the kind of fire brigade, but the immune system is a normal part of human physiology. It's always doing its thing. It's involved in tissue repair, tissue turnover. So it's health as well as disease. You'll have a pretty good sense of how the human body works, the different organs and systems at different levels. You'll have good insight into state-of-the-art thinking in our discipline, and you'll have the ability to access really top-level discovery research. You'll know the language, you'll understand the concepts, you'll have critical analysis, data interpretation skills, basically all the stuff that you need to do as a scientist. You will learn how to do that. And you will also be able effectively to communicate your ideas, both orally and on paper. You can be the most brilliant scientific genius in the history of the world. If you can't communicate your findings in a fluent and compelling way, you kind of wasted your time. So we put a lot of emphasis on that as well. What can you do when you do a degree like this? Well, I've got some pictures here of some of our recent students. So going from top left, this is Alex, who went and joined graduate medicine program over at Imperial and is, I think, now in her final year. Has done brilliantly. This is Sophie, who went straight on to do a PhD in Cambridge and is working on actually interesting project, how the interferon response, which I've just told you about in the context of virus infections, actually is involved in the pathology of Alzheimer's and other neurodegenerative diseases. And this is Daisy, who went and joined the Government Office for Science, which is like a sort of scientific version of the civil service. Walton lived in Hong Kong. He went back to Hong Kong and started a PhD on emerging infections. Sriashi set up she did an amazing thing actually. She set up a charity in India teaching children about infectious diseases and how to avoid them. And this is Leal, who actually did a PhD in viral immunology in Oxford, but then decided that she was interested in communication and trained as a journalist and is now a freelance journalist, but she writes for New Scientist and Nature as well. So those are some outcomes. Other options are master's programs. You can go and work as a research assistant. People have gone into pharma, into consulting, into law. I would argue that you can do pretty much anything. Because the kind of skills that you get from a degree like this are very universal and very transferable. And so that comes to this important question: should you choose a very specialized BSc? Because this is specialized. It's not like a kind of general BSc biomedicine. The core content, particularly the first and second year, is certainly comparable to that. So on the plus side, as I've already said, this whole business of the process of science, scientific method, is a robust and transferable skill set. Data analysis, communication, data interpretation, those numeracy, literacy, those are deployable in almost any setting. And I do think that, and I hope I've made this case to you, that studying the interaction of the immune system with infections or cancers or whatever, the perspective that we gain from that does provide us with a framework for understanding health and disease. I would argue that all disease is inflammatory, involving immune dysfunction. And so we're quite actually at the center of health and disease. And I think our discipline is intrinsically interdisciplinary, and therefore we get perhaps a broader view than you might expect from the sort of specialist nature of the title of the degree. And I'd argue also that coming to a university like this, it's all about being taught by specialists. That's the whole point. You know, if you're going to come to a research intensive university, this is what you want, I think. But what I would counter all that by saying, don't choose this degree unless you are really, really seriously interested in research in this discipline. We do have students who join the program and then want to leave it and switch to one of the other programs, which they can do. But I think that if you have any doubt, you would be better not to come and do one of these specialized degrees, because you can always specialize later. You can always focus based on interests that you acquire through your studies. So I think if you want to do the research thing, this is a good choice. If you're just generally interested in this stuff, then you're probably better off doing a broader degree and choosing modules and specializing in that way. So what I want our students to get beyond the detail is this. I want them to grasp and really have a sense of the thrill of discovery, because that's what science is about. But at the same time, appreciate that, you know, science is a serious business. It's a rigorous process, but it's also fantastically rewarding and fantastically good fun. And I think that we can do that quite effectively over the 3 years. So these are the entry requirements. This is all on the prospectus, so I'm not going to dwell on that. It's there for you if you want it. This was last year's group of graduates, and we're standing across the road on the UCL portico, having a celebration at the end of the year. I just want to finish by giving you a more general advice. Now, what can you do right now as potential university applicants? Well, the first thing is work hard, right? It's not easy. It's an academic, intellectual exercise. Work hard, be effective, but be curious, ask questions. All science is about asking questions. So don't be embarrassed to ask questions. Get information and make this an informed choice. I mean, you're here, this is good, but visit the campus, attend actual in-person open days. We have open days coming up later in this year. And think about the course and the department as well as the university. League tables and things are very much focused on universities. Your actual experience at university is massively, primarily influenced by the department that you're in and the course you're doing, rather than the university as a whole. Choose something you enjoy. You know, you're going to be here for 3 years. If you don't think you're going to enjoy it, don't do it. Do something else. Follow your interest. You've got to be interested in this stuff, because if you're not going to thrive. You're not going to want to work hard. And we want you to enjoy what you're doing. And if you're enjoying what you're doing, you'll do well. And I think the last point on here is really important as well, which is don't ever be embarrassed or ashamed about ambition, right? You know, you're here because you have an ambition. Go with that, but make it an informed one. So get as much information as you can, because then you can make the best decision. I would also say, I'll probably be struck by lightning for saying this, but if you're at the level academically where you'd be a competitive applicant for UCL, then I would say that all of the universities you're looking at, all of the programs you're looking at are going to be fine. They're all good, high-level universities. So don't be too obsessed about one university being better than the other. Okay, so that's all I have to say. I hope that was helpful and interesting. And I will be very happy to answer any questions that you have, which I think you should type into the chat, sorry, the Q&A. Although, if you want to switch on your microphone and ask a question, you're very welcome to do that as well. And Shifa was here to answer any questions, if there are any. Let's see what happens. Okay, I don't think we've got any questions.
It doesn't look like there are any questions.
Okay, then I guess we're done. So thanks very much, everyone. Shifa, would you like to conclude the proceedings?
Yes, well, thank you everyone for coming. Hope you found it useful. Hope you learned something. Maybe it taught you something about immunology that you didn't learn before. So thank you. And hopefully you can attend some of our open days so you can get to see the campus for yourself. And I suppose that's it. Bye.
Okay, thank you everyone for coming. Goodbye.